Hello everybody, my name is Elliot Kimmel, high school teacher at Central Secondary School, and this is my tutorial on DNA replication for grade 11 biology. Now I want to talk a little bit about DNA structure before we launch into replication, so here we go. These are some images that students of mine have done over the years for me using a free graphics program. They either did it in Photoshop, which is not free, or in Serif Draw Plus. Anyhow, as you recall, DNA is made of nucleotides, and down here we can see the nucleotide is circled. The yellow part here, that's going to be the sugar. This part here is going to be the phosphate group, and this part is the nitrogenous base. Now, often when you do a nucleotide, you're going to see that the phosphate is on top of the sugar. All right, and then there's the sugar, and then there's the base. That's the typical nucleotide. So I'll say I just circle one right in here, this region. But it doesn't really matter for this purpose whether the phosphate is attached above or below the sugar. At any rate, DNA is a polymer. That means it's a long chain of molecules, and it's made of nucleotides. Nucleotides are the building blocks, and nucleotides consist of three parts. We've got a nitrogenous base, a sugar, and a phosphate group. There are four different nitrogenous bases, and therefore there are four different types of nucleotide. There's thymine, guanine, adenine, and cytosine. All right, those are the four different nitrogenous bases. So if we look over at another graphic, this one over here, you can see a nucleotide is highlighted. This part is the sugar, this part is the base, and this part is the phosphate group. Those are the three parts. Over here we can see sugar, which is deoxyribose, phosphate, and base. Note that the nitrogenous base attaches to the sugar, not to the phosphate. So in all of these models, the base is projecting into the middle of the molecule off of the sugar. You'll also notice this shape here. You've got a sharp end and then a little indentation, the round end, and then the rounded indentation. This is complementary base pairing, which implies that only certain bases can bond together. So adenine over here fits into the space of thymine. So adenine and thymine complementarily base pair. It's a hard way of saying it. All right, complementary base pairing between adenine and thymine nucleotides or bases, and complementary base pairing between cytosine and guanine. What holds these together are hydrogen bonds, and they're not illustrated here, but they would be drawn as dots or a dotted line here, as opposed to the covalent bonds, which holds everything else together. For example, in the sugar phosphate backbone, which would be over here, and another sugar phosphate backbone over here, those are all covalent bonds, but the bases are held together here, not by solid lines as I'm drawing, but by dotted lines, which represent hydrogen bonds. There can be three hydrogen bonds in the case of cytosine and guanine, but when the bonding is between adenine and thymine, there are two hydrogen bonds. Okay, and we've gone over this in class, and you can see that in various textbook images as well. So this is the genetic molecule, right? This is the molecule that codes for all of your traits, and the genetic code lies in the order of the nitrogenous bases. So in this case, if we look at the right side here, T, C, G, T, G, and that's not all of the DNA, right? The DNA would extend down that way and potentially extend up this way, but this region here might code for eye color, and therefore this region here would be considered a gene. Well, really, it's the bases, again, that are coding for the eye color. All right? If you then change one of these bases, if there was a mistake in DNA, and instead of C here, there was an A, well, that changes the order of the bases. That changes the code here from TCG to TAG, and that might code for a different eye color, or that might cause a serious genetic mutation. So genetic mutations generally involve the bases having the wrong one in there, messing up the gene. All right, the gene that codes for some particular trait like eye color, hair color, blood type, that kind of thing. I want to show you the DNA replication, flash animation that I've asked you to have a look at. So this is what it is right here. This is on my website, of course. So this is the DNA replication tutorial with the worksheets. And if you've done this, then you've already got the worksheets. And essentially what you're doing is you are going to be adding in the information in the paragraph. All right, that wasn't a very nicely drawn line at all, was it? So what we've got here, let me just go to a smaller color, change it to blue. There's an enzyme here called helicase. All right, the enzyme is called helicase enzymes. And this one breaks the hydrogen bond between the bases in DNA. It breaks the hydrogen bonds between the bases on opposite strands, and it separates the DNA into two halves. So what you can see here in the drawing is there would be hydrogen bonds right in this region here between the bases. All right, and the bases are represented by these white things. There's a phosphate group and a sugar. Now, you'll notice in this model, I've got the phosphate on top of the sugar. So one nucleotide is a phosphate sugar and the base would go right there. The complementary base would be over there. So in this top case, if this first one here was adenine, then this base is going to be thymine. And if this base is cytosine, this base is going to be guanine, etc. So we are going to look at how this reaction would occur as I play the animation. So we're going to look at helicase enzyme here. And what it does is it's going to go up and it's going to cut the hydrogen bonds, as you can see here, right? So it cuts the hydrogen bonds and it leaves unpaired nucleotides. We now have two separate strands of the DNA molecule. We've got a strand over here with unpaired nucleotides, and we've got a strand here. Now keep in mind again, if this was an A, that was the T. They were joined by a hydrogen bond, but the enzyme here, helicase, has cut it. Now think of an enzyme just as a big globular molecule, or like some kind of a Pac-Man thing there, you know, going through, there's the teeth there, going through, chopping up things. So this molecule had come up here and cut the hydrogen bonds, separating this side from this side, A from T, separating C from G, and so on. So we now have two unpaired strands of DNA, and what we want to do here is we want to replicate them. So we're going to go to the next frame here, and it introduces another enzyme. The enzyme here is called DNA polymerase. Now, it's called that because it's going to act on DNA. Remember, this is DNA. And the word polymerase means it's acting on a polymer. Now, a polymer is a chain of molecules connected together. That's like poly for many. Polymer means many molecules connected together. What's going on with that thing? Get out of there. Hang on. Okay, back to this. All right, so DNA is a polymer of nucleotides. There's a nucleotide down there. Each of these is a nucleotide attached to the next nucleotide, attached to the next one, and to the next one in a long chain right here. This is called a polymer. So the enzyme DNA polymerase is going to come in here and create a new polymer, a new chain. So this base is sitting here without its nucleotide. Nucleotide was over there, right? There's an enzyme in the nucleus of the cell. This is where this is happening, right? In the nucleus during interphase, late interphase. This enzyme, the second one in the sequence, is called DNA polymerase, and it finds nucleotides that are just floating around. So, for example, here is your cell. Here is the nucleus, and inside the nucleus is your DNA. So let's draw a little strand like that. And there are many nucleotides, free-floating nucleotides. There are adenine nucleotides and thymines and cytosines and guanines, and they all come from your food, whatever you've eaten, whether you've eaten a hamburger and you've got nucleotides from a cow, and you've got nucleotides from the lettuce and from the pickles, from the plants. All of these are nucleotides because all living things have genetic material like DNA, and so you eat them and you break those food particles down into nucleotides, which you then use to build your own DNA. They go into your DNA. So there are lots of free-floating nucleotides in the nucleus. And this enzyme recognizes DNA has been split and says, oh, I have to create the other side. So it's going to take this nucleotide and it's going to click it on here. And of course, if this wasn't a T, let's say, then this would be an A. And then it's going to grab another one and it's going to click it on there. And so if this was a C, then it's going to click on a G. And it's going to move up on this side and it's going to add the correct complementary nucleotide onto there. Another DNA polymerase, because there's more than one in the cell, is going to come along and it's going to see this side. Now, this side was an A. If this was a T here originally, and it clicks an A on here, this was the partner, the complementary base. It was an A. So it's going to come up here and it's going to click a T on there. And then it's going to click the correct one on there, and then on there, and then on there. And it's going to build that half, and we will have 2 DNA molecules. Now let me erase that and go through and play the animation. So there it's adding nucleotides on the left, and then it adds nucleotides on the right. And in the end, you now have 2 DNA molecules that are actually identical. And you'll see that when you go through the actual tutorial. All right, let's move on and look at the last part. Actually, just before I do that, just notice one thing here. DNA helicase cut the strand at first, or just helicase, cut the hydrogen bonds, separating them out into 2 halves like this. All right, the ladder-like drawing. It cut between the bases, right up the middle, and you had 2 halves. DNA polymerase attached these new nucleotides. So we now have 2 ladder-like DNA molecules. But you're going to notice this black thing in here. This represents a bond between the sugar of the upper nucleotide and this thing in cyan or blue or green here, the phosphate there. There's a bond between the sugar and the phosphate, the sugar and the phosphate, the sugar and the phosphate, all right, to make up the sugar-phosphate backbone. And it exists in the original other half because these 2 were originally joined together, right? These are the new nucleotides added, and they are bonded here. Complementary base pairing is occurring here, hydrogen bonds, all right? So that is a bond. They are held together. But the sugars and the phosphates are not connected along here. These were just free-floating nucleotides clicked on, glued on, there and there and there, but they're not bonded here as they are here. So the final step in DNA replication is to create the bond there. So let's have a look at how that works. So we have our third enzyme, and our third enzyme is called ligase. That's another horrible line there. I'm going to circle this, try to circle it. There we go, ligase enzyme. All right, ligase is the enzyme that creates a bond right there where it says no bond here. It's going to put in a bond. It's going to glue the sugar here to the phosphate there, and it's going to glue this sugar to this phosphate so they're connected as they are here. So let's watch how that could potentially work. Okay, it's clicking in the bond. There we go. At that point, the sugar-phosphate backbone exists on both halves of the DNA. We've got one polymer of DNA, one strand of the DNA, and we have the other strand of the DNA with the ladder-like rungs in between, the nitrogenous bases, and that is a DNA molecule right there. We have another DNA molecule, and it's exactly the same right there with the sugar-phosphate backbone bonded together and hydrogen bonds holding the nitrogenous bases together. This is our characteristic chromatid joined to another chromatid. So if I draw this right here, right, this one here, number 1, this DNA molecule, that's this side. This DNA molecule here is this one. So if you looked inside, it would like go around like a DNA. I can't draw a DNA, but you know, can you imagine that mess, right? And there would be a centromere, a structure here, sort of holding it together right there. So we end up with 2 identical DNA molecules because of the way DNA replication works. All right, and that is how the tutorial works for the enzymes. The next part of the DNA replication tutorial is essentially just dragging and dropping and trying to do it yourself. So you could switch on the help to see what you have to do, but I'm just going to turn that off Actually, I'm going to turn that on because I want to show how the base pairs work. So what I've got here, let me do this first, is I've got a nucleotide. So I'm going to come down to this one, right? This has a phosphate, a sugar, and a nitrogenous base represented by this white square right here. Sugar, phosphate, nitrogenous base. This one has a C, a cytosine. So in this case, we would need to drag up, let me get rid of the help actually, and get rid of that. We would need to drag up the correct letter. So if this is cytosine, this would be guanine. And if this is guanine, this is cytosine. Thymine pairs with adenine. Cytosine pairs with guanine. And adenine pairs with thymine. All right. That shows you a short section of the DNA molecule right here. All right. Phosphate sugar, guanine, phosphate sugar, cytosine. Of course, there's a hydrogen bond right in there waiting to be broken by the enzyme helicase when it's time for DNA replication. So in this case, what would happen is helicase would come in here and it would sort of slide up through the middle and it would separate these bases from each other. So if this is cytosine, guanine has been pulled away from the cytosine by breaking the hydrogen bonds. And if this is guanine, cytosine will go in here. Whoops, let's grab that, etc. So you could see how the rest of these could be filled in. So these are the individual strands of nucleotides separated from each other. And then in the next step, DNA polymerase is going to come along and click on nucleotides. So I'm just dragging the enzyme up anywhere saying, okay, that's what it's going to do there. So, for example, if we see a cytosine here, the enzyme DNA polymerase would grab a guanine nucleotide, bring it up, and a hydrogen bond would form there, and the DNA polymerase has done its job. Oh, come on, get off there. Okay. There we go. Guanine, we're going to put a cytosine there. Whoops, I skipped ahead a little bit. Thymine's going to go there. Another guanine is going to go there. And an adenine is going to come over here. Now, these were new nucleotides, free nucleotides that were floating around in the nucleus that this enzyme grabbed and clicked on. Now, if it grabs the wrong one, once again, like if it tried to put the A there, that's a mistake, right? That's a genetic mutation. That's not going to work properly, and that could lead to problems down the road. All right, so we get that one. And, of course, the same thing would happen on the other side. Now, these ones are left blank because you were supposed to drag in the letters. So if cytosine was there, this nucleotide was guanine. This one was originally over here. So there it is there. So DNA polymerase would see that G, and it would bring in a cytosine. In the end, you would get two identical DNA molecules, and that would be the process of replication. The final step, of course, is for ligase to come in and to create the sugar phosphate backbone, and it would just create a bond here, and it would create a bond here, and it would create a bond between all of the nucleotides along the side. And so you should have this worksheet, and you fill in all the information, and you would see the final product. You'd see that this one and this one are identical. All right, and that's basically how the tutorial was to go.